So we're now thinking about enzymes. And an enzyme is a biological catalyst. And what a catalyst will do in chemistry is it will lower the activation energy for a reaction to take place. In other words, a catalyst will increase the rate of a chemical reaction. It will facilitate and increase the rate of a chemical reaction, making a chemical reaction more likely to occur. So enzymes are biological catalysts. They facilitate biochemical reactions. They control and regulate biochemical reactions. Activate, control the rate of, basically they govern all the biochemistry that's going on in the body. And you can always tell enzymes because they end in ase. So for example, a protease is going to digest proteins. An amylase is going to digest carbohydrates. A lipase is going to digest fats. But as we'll see, there's other types of enzymes as well. And enzymes are proteins. They're usually globular proteins in nature. And the key thing about an enzyme is it's not used up in the reaction. So this is true for a catalyst as well. So an enzyme or a catalyst, and we're thinking about enzymes, they are not used up in the chemical reaction. They are not part of the reaction. They just facilitate the reaction, regulate the reaction, increase the rate of the reaction. And what enzymes do is the enzyme will actually work on a substrate. So there'll be a substrate that's acted on by the enzyme, bringing about some biochemical process, and the end result will be a product. So the product is generated by the action of the enzyme, or the substrate is facilitated into the product by the action of the enzyme. So it's substrate into the enzymic system, product out of the enzymic system. Now enzymes are proteins. They're usually globular proteins. And proteins are long chains of amino acids, and they fold together into a secondary structure, and they fold even more times into a tertiary structure. Proteins have this complex protein folding pattern determined by the primary sequence of the amino acids. And what this means is that proteins are able to generate a molecular architecture. They have shape. They generate physical shapes with electrical properties, but they are nonetheless physical shapes. And enzymes have this physical shape that they generate as a result of this complex folding of the protein, and it's called the active site. So enzymes have an active site, and the active site is where the substrate binds into, undergoes its chemical processing to be released as a product. So this active site is actually where the activity goes on. It's the active site of the enzyme, this very specific particular shape. Now the examples I've just given of enzymes are actually digestive enzymes. But there's two main ways we could look at enzymes in the body. There are the enzymes inside cells, the intracellular enzymes, and there's the extracellular enzymes. So throughout the body there are intracellular enzymes. So in each cell of your body there's a milieu of unbelievable complex biochemistry going on, with numerous biochemical reactions going on in different parts of the cell, and all of these are facilitated by enzymes. They are accelerated by enzymes, they are controlled by enzymes. Without enzymes there is no life, because all this biochemistry that generates life is facilitated and controlled by enzymic systems. And actually in clinical practice this is quite useful, because when a particular cell dies, the enzymes that were inside that cell will be released because the cell membrane breaks open. So cell death, or even some cell injury, will release enzymes into the body fluids that were intracellular enzymes. And this is quite useful because we can do blood tests, look for these enzymes, and from that we can work out which part of the body is injured. So creatinine kinase might indicate a muscle or heart injury, or AST, aspartate aminotransferase, might indicate liver damage. Or there's quite a few enzymes that you'll come across in practice, but they are enzymes that indicate damage to particular tissues. Or sometimes we can even look for isoenzymes, because an isomer has got the same chemical formula but a slightly different structure, and there's different isoenzymes in different organs and tissues of the body to perform particular biochemical activity in that particular area of the body. But it's useful, so we can look for these enzymes as markers of particular tissue damage, because they've been released from the cells of that particular tissue when that tissue was injured, insulted, or indeed necrosed and killed. So it's quite useful in clinical practice. But extracellular enzymes, which we're getting around to thinking about, are used in the gastrointestinal tract. So these are usually secreted into the lumen of the gastrointestinal tract, where they are breaking down food components. And the aim here is to break down complex food molecules, which are usually insoluble. Complex food molecules to be broken down into soluble products, because we need soluble products. Basically it means smaller molecules which are water soluble, usually water soluble, some can be fat soluble. But breaking large molecules down into smaller molecules which can be absorbed into the bloodstream or into the lymphatic system. So most digestive enzymes are working within the lumen of the gut. Those on the enterocytes, the very final stage of digestion, are actually operating on the cell surface membrane of the enterocyte itself. So they're either in the lumen of the gastrointestinal tract or very close to the gastrointestinal tract. And that brings us on to another way we could look at enzymes. We could think about enzymes as being anabolic or catabolic. So anabolism, anabolic processes, are building small molecules up using energy into bigger molecules. That would be an anabolic process, as goes on, for example, in the synthesis of protein to make muscle, where we need to build up larger molecules, in this case protein molecules from smaller molecules, which would be the amino acids. So that's an anabolic process. It's a building up, making bigger molecules. Some enzymes will facilitate these anabolic processes. Other enzymes will facilitate catabolic processes. Catabolic processes are to break big molecules down into smaller molecules. So the digestive enzymes we were considering are going to be catabolic, because they're breaking things down into smaller amounts, into smaller molecules, making them more soluble. Now, enzymes are very particular, very fussy, very specific, and they're very specific to the substrate that they can enzymatically convert into a product. So enzymes are substrate specific, and this is because the active sites that form a particular molecular architecture are a highly specific shape, and they have highly specific electrochemical properties on their surfaces as well. So they will only accept one particular type of substrate. So an amylase, for example, thinking about digestive enzymes, an amylase will only accept carbohydrates. It won't accept fats, it won't accept proteins. It will only break down carbohydrates into simpler sugar and disaccharide units. They're highly specific to substrate, but as well as that, they're highly specific to temperature. So at lower temperatures, the substrate, imagine that's the substrate there, and this is the active site of the enzyme. So the two have to come together. The substrate has to get into the active site of the enzyme. Now at lower temperatures, kinetic theory tells us there's less molecular vibration in a fluid. And as temperatures increase, the molecular vibration is going to become more intense. So that's a cold substance, and that's a warm substance, more vibrations. And the more vibrations there are, the more moving around, the more likely that is to happen, that the substrate will fit into the active site of the enzyme. So activity of the enzyme tends to increase with increasing temperatures, but then it reaches an optimum point because as temperatures rise, that's going to affect the chemistry of the active site. And as temperatures rise, the active site can be denatured. It can go out of shape, which means the substrate can no longer fit in because there's been denaturing of the active site as the temperatures become higher. So what we find is enzymic reactions get faster and faster as the temperature increases, then they reach an optimum, and then they can drop off quite quickly due to denaturing of the complex molecular architecture that is generated by the active site. And they're also very specific. Enzymes are very specific to pH, to the acidity, because the pH also has the potential to change the nature and the shape of the active site. And again, alterations in pH can lead to denaturing of the active site. Now, it's probably well known that the pH of arterial blood is 7.35 to 7.45, so you'd expect a lot of body enzymes in the cells to work at that sort of those sort of pH levels. In the gastrointestinal tract, it's slightly different. You get some enzymes working in the stomach where the pH is very low. You get some enzymes working in the small intestine where the pH is high. Low pH being acidic, high pH of course being alkaline. Or you get amylases working in the mouth where the pH is relatively round about neutral levels as well. So it's not that enzymes work at one particular pH, but specific enzymes work better at a specific pH, but that's dependent on the particular biochemistry of the enzyme itself. And indeed the rate of reaction can also be increased if there's a higher concentration of enzyme, if there's a higher concentration of substrate, and also it helps if the product is continuously moved as well. Otherwise the product tends to get in the way of the substrate fitting into the active site of the enzyme. So that's the little conceptual introduction to the nature of enzymes. Next we want to look at a little bit more about this interaction between the substrate and the active site, a little more about the specificity of enzymes, and then we'll need to go through the specific enzymes that have specific roles within the gastrointestinal tract in this role of digestion, breaking down big insoluble molecules into small soluble molecules ready for the process of absorption. So as an example of how enzymes work, we're going to look at this enzyme here. And this enzyme here is sucrase. And sucrase will break down sucrose into its constituent molecules. And a molecule of sucrose is just ordinary cane sugar that we get everywhere. We also get it from sugar beets, the most common sugar that we use. Table sugar is sucrose. But that's going to be one molecule of glucose and one molecule of fructose. That's what the two molecules are that compose this disaccharide. So there's a monosaccharide glucose unit, there's a monosaccharide fructose unit forming the disaccharide sucrose. Now with random movement, this can fit into the active site of the enzyme like this. So this molecule is now fitted into the active site, and the sucrase enzyme, what it will actually do, it will put a stress on the bond. So the sucrose fits into the sucrase, that's the sucrase, that's the sucrose. And the sucrase enzyme is going to put a stress on this bond here between the glucose component of the sucrose and the fructose component of the sucrose. This is actually called a glucoside bond, it's glucoside bonding. But it's now under stress. So this bond is now under stress. Now, these reactions that break down food products into smaller components are all hydrolysis type reactions. Hydro, water, lysis to break up. So we need to add water to this. So water is going to be added. So strain is going to be put on this glucoside bond by the sucrase enzyme. That's going to allow water, facilitate the addition of water to the molecule. And then what will happen very quickly is that the enzyme will result in the breaking up of the two. And we now have an individual molecule of glucose and an individual molecule of fructose. The digestive process has taken place, and they're now free to be absorbed. Physiologically, this would actually happen on the brush border of the small intestine, mostly the ileum, that brush border where a lot of this final stages of digestion take place. So we now have a digested two molecules as a result of the enzyme. Now, firstly, we notice the enzyme is free. The enzyme is now free to get the next sucrose molecule. The enzyme has not been used up. The enzyme has not been changed. But water has been added, and specifically the glucose has received an OH group, and the fructose has received an H, a hydrogen atom. But that digestive process has now taken place, and the enzyme is free to... They're going to be absorbed, so they'll now be absorbed through the gastric mucosa because they're water soluble, through the villi, into the hepatic portal vein and off to the liver. And the glucose can just go straight through the liver, but the fructose will be converted into glucose the first time it goes through the liver. So the only sugar actually going into the blood will be the glucose. So this enzyme is now free to do the next molecule that comes along as they go off to be absorbed. And the rate of reaction for this kind of process is thousands of substrate molecules, thousands of these substrate molecules per minute. So this is a remarkably fast process going on really quickly. Because if a solution of sucrose is left in a sealed container, it breaks down into glucose and fructose extremely slowly, barely measurable. But in the presence of even a small amount of the enzyme, in this case sucrase, the rate of breakdown is accelerated by millions of times. It's millions of times faster. So just to review a few points, enzymes are proteins which act as a biological catalyst, facilitating and, as we've seen, greatly increasing the rate of reactions. And enzymes speed up reactions without being used up in the reaction itself. So this was completely free to go on and metabolize or to digest the next substrate molecule. So what's happening is there's a substrate. In the case we looked at, the substrate was sucrose. It was broken down into the product, which was glucose and fructose, and the enzyme facilitating that process was sucrase. But this is the way it works. The substrate is converted into the product, but only under the influence of the enzyme. Now it's interesting to think about how enzymes are naming enzymes. And the principle often is it's the substrate or the substance being acted on, and then an A's on the end. So for example, sucrose is digested by sucrase. So the A's indicates that it's an enzyme, sucrase. Now, having said that, there are some examples of enzymes which are named before this convention became well recognised. So it's not a universal rule, but generally enzymes end in A's. So it's the substrate, sucrose in this case, followed by A's. So this was a sucrase enzyme. And enzymes can be classified by the type of chemical reaction they catalyse. So the example we looked at was sucrose going to glucose and fructose by the action of the sucrase and the adding of the water. And we noted that the glucose ended up with an OH and the fructose ended up with an extra H, but they broke down into separate units. So these are both separate units at the end of this process. Monosaccharide units. And so this reaction, as we can see, this reaction here involved adding water. So this reaction involves adding water, a water molecule, to break the chemical bond. And that's why it's a hydrolase. It's a hydrolase enzyme. So these are, all digestive enzymes are actually hydrolase. So hydro is the prefix meaning water, and as we know, ase means enzyme. So they're hydrolase enzymes which break down by the addition of water. And all digestive enzymes are actually hydrolase type enzymes. Now, just before we go on to look specifically at the individual digestive enzymes, I think we'll just notice one more thing about enzymes. There can be anabolic enzymes or catabolic enzymes. So these hydrolase enzymes in the digestive system, are they going to be anabolic or are they going to be catabolic enzymes? Well, the answer is they're going to be catabolic, because a catabolic reaction is breaking large molecules down into smaller molecules, whereas an anabolic reaction is going to build smaller molecules up into a bigger molecule, such as synthesizing a protein. And this is interesting because enzymes can be intracellular or extracellular. So intracellular means the enzyme is working in the cell, whereas the digestive enzymes are going to be extracellular. They're working in or very near the lumen of the gastrointestinal tract, so they're actually working outside of the cells. But many enzymes are intracellular. They are working within the cell, so within a cell here. And in a cell there are untold numbers of complicated chemical pathways going on. So I'm just going to make a really simple one. So sometimes we need product A to be converted to product B, to product C, to product D, for example. And these stages in this biochemical processing are all going to be facilitated typically by different reactions. So that's going to be facilitated by AAs, because A is the substrate. So that would be AAs there, broken down by AAs. This next step will be facilitated by BAs, because B is now the substrate, and A's means enzyme. That step will be facilitated by CAs to get the final product we want, which in this case is D. So these would be intracellular enzymes working within the cell. But in the next video in this series, what we want to do is go on and look at the specific extracellular catabolic enzymes involved in the processes of digestion. That is the process of breaking down complex food molecules, which are typically insoluble, into highly soluble, smaller products of digestion fit for absorption into the bloodstream or the lymphatic system, in order to be used as nutrients and providers of energy in the rest of the body. Now, one of the important concepts about enzymes to grasp is that they are highly specific. They are specific molecules designed for a specific purpose. And specifically, that they are specific to the substrate that they're going to act upon, the temperature at which they are going to act, and the pH of the environment in which they are acting. So first of all, let's think about the nature of the substrate. Now here we have our enzyme with its highly specific molecular architecture. And here we have the active sites. These are the active sites. And we'll suppose that this is the sucrase enzyme we have been considering. Now, here's a molecule coming along, and this molecule might be, for example, an amino acid. And because of kinetic theory, we've got movement of the enzyme in the fluids and movement of the amino acids, two amino acids here, small protein, part of a polypeptide. And they're moving around, and they might just happen to come together like that. Now, can we see that there's no reciprocal shape between the substrate, or the potential substrate, and the enzyme here? So that's just going to wander off into space again, because there's going to be no bonding, there's going to be no substrate enzyme complex formed, there's going to be no temporary bonds, because everything's in the wrong shape. All the electrochemical components of the enzyme and the substrate don't match up, and the physical shape doesn't match up. The whole thing's completely hopeless and doomed from the start, so that will just wander off into space like that. But let's suppose the correct substrate comes along, in this case sucrose. That's now going to, again by random movement, it's going to sometimes fit into there. Oh, that looks a bit better, doesn't it? So now we've got a perfect match between the substrate, this is the sucrose molecule, and the sucrase enzyme. We now have a perfect match between these two. So we see that enzymes are specifics for substrate. That one simply won't do, but that one fits perfectly. So high specificity in terms of the substrate that can be enzymically catalyzed by the reaction, by the enzyme. Now enzymes are also highly specific to temperature. So let's see what's going on here in terms of specificity for temperature. Now you're probably familiar with kinetic theory. Now what kinetic theory does is it says any particle, it doesn't matter what it is, it could be my lens cap here as long as it's a microscopic particle. These microscopic particles are going to be acted on by molecules in the fluid. So in a fluid there's going to be lots of molecules, probably mostly water. Lots of molecules like this all around. And as the temperature increases, the energy of those molecules is going to increase. So if we take any one molecule, it's going to be vibrating more with increasing temperatures. And if we increase the temperatures more, the vibration is going to increase. So for any individual particle, let's suppose this pen lid is an individual water molecule, that's going to be vibrating at a rate which is determined by the temperature. That's what heat is, it's molecular vibration. So there it would be very cold because it's still. But as it gets warmer it moves more, and as it gets hotter and hotter it moves more. So heat is molecular vibration, that's what it is, and it occurs in a chaotic direction. But if we have larger particles here, like this, still a microscopic particle but larger, then that's going to be acted on by water molecules from that side, and from that side, and from that side, and from that side, and from that side. But remember, these water molecules are themselves acting in a chaotic way. So what will happen is, at any one moment in time, you might have several water molecules hitting it from that side, more than you have hitting it from that side. So what this means too is that these more macroscopic particles, not macroscopic, but larger molecules, are also going to randomly vibrate. This is called Brownian motion, and it's caused by the vibration of the individual water molecules just hitting this in an irregular way, causing this movement. And it's the same because, of course, the substrate is going to be in a watery medium, so that's going to be vibrating. And as temperatures increase, it's going to be vibrating more. And of course, it's the same for the enzyme. That's going to be vibrating as well. So you can see if these are pretty still, there's not much chance they're going to come together. But if there's more vibrations, if there's more molecular vibration going on, then okay, sometimes it's not going to fit, but other times it's going to come in the right direction. So increasing temperature increases the probability of the substrate hitting the right active sites within the enzyme and forming the substrate enzyme complex. And then what we actually see, if we think about this graph, this explains what's happening here. So what we have here is we have enzymic activity there. So that's 100% when the enzyme's working as much as it possibly can, and that's 0 when it's not working at all. That's 50%. And here we have the temperature. So this is an enzyme which is working at physiological temperatures. So 0 degrees centigrade, 20, 40, 60 degrees centigrade. And we find out that as the molecular vibration increases with the kinetic theory and the Brownian motion of the substrate and the enzyme, that the probability of the substrate enzyme complex forming is going to increase as the temperature increases because there's more molecular vibration. And then it's going to reach a peak point here, and this is a physiological type enzyme, probably working in a body cell, that's reaching optimum at about 38 degrees centigrade. So that's good. But then if we carry on increasing the temperature, although it's true if we carry on increasing the temperature, you're going to get even more Brownian type motion. But the trouble is, as we increase the temperature, because this active site is generated by very complex folding of the protein, what will happen if we increase the temperature further is we will get alteration of the physical shape of this and therefore the electrical chemical properties of this active site. It will physically change shape and chemically alter. And that is called denaturing of the active site. So they will be denaturing of the active site. So if we increase the temperature a little bit, we're going to get some denaturing of the active site. But then if we increase the temperature more, we get very rapid deterioration of the active site. And it becomes unrecognisable. Therefore, the shape of this is going to change completely, so it won't fit in. So with increasing temperatures, we get denaturation. And that's irreversible. This protein is then damaged forever as a result of heat. In burn injuries, we call this a coagulation necrosis. We get coagulation of the proteinaceous tissue and death of the proteinaceous tissue in a coagulation necrosis, which is what causes the burn injury essentially. It also happens when you fry an egg. When you put in the egg in your frying pan, it's the albumin, the white part is clear, but then it goes cloudy as the proteins denature. So we get denaturing of this and therefore rapid drop off in enzymic activity with increasing temperatures. So the enzyme is going to work optimally within a fairly narrow range of temperatures. Now the other specificity we mentioned about enzymes was pH. So we've talked about specificity for substrate temperature. Now we're thinking about the acidity or the alkalinity of the environment. So again, we notice that this axis is referring to enzyme activity, 50% of activity, 100% of enzyme activity where it's working as quickly as it can. And here we have the pH along the bottom, so from a theoretical pH of 0 up to a neutral pH just there, and then a very alkaline pH up here at 14. And here we have an enzyme which is working at physiological pHs. You might remember the pH of arterial blood is 7.35 to 7.45. So this particular enzyme is working within that kind of range. But we notice that the range is relatively narrow. So the temperature graph actually was a bit spread out, wasn't it? There was potential activity between these 2 points. But with pH it's much more specific. This, of course, is why acidosis or alkalosis is such an important clinical finding. It's very serious if someone becomes acidotic or if they become alkalotic, because all of the body enzymes are no good. All of the somatic body enzymes, the intracellular enzymes, are nearly all attuned to work at these physiological pHs. And if that moves very much in either direction, either towards acidosis or towards alkalosis, then enzymic function in the cells will just not occur, and therefore there'll be no life-giving biochemistry. So quite a narrow range really for pH. And again, the rationale for this is much the same as it was for the increasing temperature. So at reduced pHs, the shape of the active site of the enzyme is going to be denatured. And it's the same with increased pH in alkaline environments. It's going to be denatured as well. So alkalosis or acidosis can cause denaturing of the active site of the enzyme, therefore meaning it's no longer the correct shape to accept its substrate. Therefore the enzyme will lose its ability to function in this physiological way, forming the substrate enzyme complex.